Thank you very much, Simone, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, so hello everyone. First of all, thank you very much, Kate, for inviting me to present the Build a Cell seminar today. So just briefly introducing myself, my name is Simone Giaveri, and currently I'm a postdoctoral fellow at the Max-Planck-Institute for Terrestrial Microbiology in Marburg, Germany, and I work in the department of Tobias Erb. So today I decided actually to focus this presentation on my postdoctoral work that I carried out here at the MPI Marburg. But in the end, I also decided to connect this presentation with my previous PhD work that I carried out at the EPFL in Lausanne, Switzerland. And also in the end, I decided to introduce a little bit what are my current and future research interests. So you can see actually what I'm really passionate about at the moment and that I will carry out as an independent group leader. So after this brief introduction, let's finally talk about science. And I would really like to start this presentation by talking about synthetic cells. So what are synthetic cells? Actually, this is a question that is not super easy to answer. And for this reason, a few months ago, in October 2024, we met in Shenzhen, China, for the first SynCell Global Summit together. And you can see both me and Kate there. It was a great time in the picture. And in this summit, we discussed together what are synthetic cells, as well as what are the current main challenges and also future opportunities in our field of science. And despite the modular approach we mostly use all over the world in our labs to try to attempt to build synthetic cells, the accent on the definition of synthetic cells during the meeting was on the ability to self-sustain, to replicate, and ultimately to evolve. And so now that we have seen what is a synthetic cell, then I wonder why should I build a synthetic cell? And then, well, I can answer to this question with many different answers. And the first one is that I'm a very curious person. But then I think also this type of research can allow us to understand some principles of life that would be very complicated to discover if we were to work in vivo due to the complexity of biological systems. And in addition to this, as in the end, I'm a materials engineer, this type of research can allow us to engineer advanced biomimetic systems that can be used for a plethora of different applications, ranging from advanced materials and materials engineering and towards also the biomedical and medical field. And so up to now, we have seen what are synthetic cells, a few reasons why it should be interesting, could be interesting to build synthetic cells. And then now let's see how to try to build synthetic cells. And so in my work, I mainly focus on the so-called bottom-up approach. That is, I try to integrate together non-living components such as proteins, nucleic acids, lipids, and also small molecules into systems whose composition is well defined. And if the composition of such systems is well defined, then this is a big advantage because it allows me also to control the properties of these systems pretty well. And but you can also, of course, imagine that if I assemble by the bottom up the components I need to obtain a specific function of the system, then it means that I integrate together only the components I need to achieve these functions. And so we can also call these synthetic cells minimal cells because they are composed of the minimal elements to make the system work. But you can also imagine very easily that this type of, this field of research is highly interdisciplinary. And so how did I actually get passionated? How did I jump into this field of science? So the answer here is because also my CV is highly interdisciplinary. Originally by training, I'm a materials engineer from the Politecnico di Milano, the NATA department. And then I had the chance to move for a PhD at the EPFL in Lausanne, Switzerland, also for a PhD in material science, but that in the end, it was really in reality at the interface between material science and cell-free synthetic biology. And for this reason, I was advised by Francesco Stellacci for the material science part of the PhD and by Sebastian Merkel for cell-free synthetic biology part. And I have to say that I really got passionate being in Sebastian's lab about bottom-up synthetic biology. I got some, I think, cool ideas in mind and decided that the best option for me was to go for a PhD for a postdoc in cell-free bottom-up metabolism. And for this reason, I actually decided to join the department of Tobias Erb at the MPI Marburg, where currently I am, because I think that Toby is one of the world leaders in this field. And in this regard, I really have to thank Toby as well as the Max Planck Society for the support during the last years for my research, as well as EMBO for funding my postdoc. So after this short introduction about my CV that I think could also be interesting because you can see then where the research I'm gonna show later on during the presentation has been carried out in the different European countries. And so let's jump back to science and specifically to minimal cells that we have just introduced before. And so, as you know, a lot of work has been already carried out in the field of bottom-up synthetic biology to try to assemble minimal cells from non-living components. And more in detail, this work has been carried out mainly to try to mimic individual functions of living cells in vitro, such as compartmentalization, communication between different compartments, energy supply and metabolism, as well as DNA replication and protein production, and also growth and division and motility. But today now I would like already to highlight cell-free metabolism and cell-free protein production because we are gonna focus on these two biomimetic functions a lot during this presentation. But as I mentioned before, in the end, I'm a material scientist, and as a material scientist, there is a property typical of living system that I do particularly admire, and it's the ability of living systems to autonomously self-regenerate and repair. In this regard, I think that keynote work has been performed by two colleagues of mine back at the time I was in the Merkel lab at EPFL, that are Barbora Lavikova and Nadanella Kunakorn, who were able to pioneer self-regeneration in vitro by using transcription and translation in a microfluidic reactor. And what they did is that they were able to assemble a special pure system that could follow the instruction encoded into the DNA using the anabolic tool of transcription and translation, and it was able to self-regenerate once it was supplied with the necessary building blocks, such as amino acids. And this work for me has been really inspiring. It has inspired my research of the last years. But when I look at nature, what I see is that actually living cells are open systems. And so they are actually able to autonomously make use of energy sources as well as matter in the forms of building blocks and also precursors, so to produce the macromolecules and molecules they need. And this is through metabolism, that is a complex network of chemical reactions that happen in living cells, and that provides living systems with the energy and building blocks that are necessary for life. And so the main research question I had when I started my postdoc at the MPI Marburg was to see if I could build a metabolic and genetic linked in vitro network that I like to shorten as MGLN, that again is able to take the information stored into DNA using the anabolic tool of transcription and translation in a pure system, but this time it is able to self-regenerate by at least partially producing some of the needed building blocks for regeneration through its own metabolism. And in my case, this means from CO2, that is also inorganic carbon. So how to do this in practice? Well, the idea was to start from the so-called crotonyl coenzyme A, ethyl-malonyl coenzyme A, hydroxybutyryl coenzyme A cycle that we normally shorten as CATCH, and it is cell-free. It means it works outside cells. It is synthetic. That means it doesn't exist in nature. And a CO2 fixing system that was invented a few years ago by my supervisor, Tobias Herb. And it starts from propionyl-CoA. It is able to fix 2 CO2 molecules per turn by producing glyoxylate, that is a C2 output, and also regenerating propionyl-CoA. And the idea was to integrate this system together with a very special pure system that does contain all the necessary elements for transcription and translation, apart from the amino acid glycine. And the idea was to convert the glyoxylate coming from CO2 into glycine by using an aminotransferase enzyme, so to activate translation in the pure system. So this looks pretty cool on paper, but actually how to make this work in the lab? Well, the first step was actually I tried to establish a so-called delta glycine pure system, that is a pure system that depends on the externally added glycine to perform the transcription and translation of a EGFP gene that you can see sketched on the right-hand side of the slide. And you can see that if there is no glycine in the system, then I could not record any fluorescence for the transcription translation of GFP. But as soon as I provided the system with the same concentration of glycine as the other amino acids, that means 300 micromolar, then I was able to record a nice fluorescence signal into the plate reader. And this was already pretty nice, but I was also curious to see if I could identify a range of concentration of glycine for which I could find a linear dependency between glycine concentration and the fluorescent plateau achieved by transcription translation. And so you can see now in the plot that every time I was doubling the concentration of glycine in my system, ranging from 10 micromolar glycine to 150 micromolar glycine, every time I was doubling the concentration of glycine, I was obtaining a 2 times higher fluorescent plateau. And this, I think, was pretty cool because it meant that I could control the system really robustly and really precisely. And so after this result, I moved directly to I think, the main point of this project, that was the selection of the best aminotransferase candidate, so to convert glyoxylate into glycine. And why is this like important point? Well, because this reaction from glyoxylate to glycine should happen in the presence of all of the other amino acid residues. And so you don't want to produce glycine by depleting any of the other residues. You want to produce glycine out of CO2. And so after a few trials, we identified the glutamate glyoxylate aminotransferase GGT1 from Arabidopsis thaliana. Why? Because this enzyme was able to produce about 200 micromolar glycine after about 15 minutes without modifying the concentration of any of the other amino acid residues. And after this important result, I wanted to move immediately to the delta glycine pure system that now you can call also delta glyoxylate if you want pure system, and substituted glycine in the same experiments I commented before with glyoxylate and keeping constant the concentration of the transferase enzyme. And so you can see now that basically the system behaved exactly as happened before. And I could again identify this linear range of dependency between now glyoxylate and fluorescent plateau for the TXTL of GFP in the same range of concentration, that is between 10 micromolar and 150 micromolar glyoxylate, keeping constant the GGT1 concentration. And so after this, it was really a time to start combining the two layers of metabolism and genetics together. And the first step I did was to try to supplement the pure system one by one with the components of the cat cycle and measure the performances of transcription translation in the pure system by using all the time this transcription translation of EGFP gene sketched here at the top of the slide. And you can see that first I added some salts and then ATP, formate, coenzyme A, NADPH, and also then the mixtures of the enzymes of the catch, one by one. And then what we did is that I also then switched to add the whole cat cycle into the pure system. And in this case, what I decided to do was to keep off the cat cycle. That is, I omitted propionyl-CoA, that is the starting metabolite, to make this work. And I instead activated the pure system by providing the system with a DNA template, again encoding for the green fluorescent protein. And I also supplemented the system with 300 micromolar glycine. And you can see on the right-hand side of the slide that once we're using a 5 times diluted cat cycle into the pure system, then the performances of the transcription translation, the pure, were actually pretty high. And then what I did exactly the opposite. And so what I did, I decided to take the 5 times diluted cat cycle and activate it into a pure system that was instead deactivated. And so in this case, the cat cycle could start turning because propionyl-CoA was fed to the system, but the pure system was not supplemented with any DNA template, and so it couldn't work. And the goal of this experiment was to see how much glycine could a 5 times diluted cat cycle produce in the presence of all the elements of the pure system. And I was very positively surprised to see that after a few trials, I was already able to produce about 110 micromolar glycine, you can see here on the plot, over a negative control that is about 1 order of magnitude lower. And this result was pretty good, actually, very good. Why? Because 110 micromolar means that we are inside the linear range I commented on before. But at the same time, I also wanted to see if I could actually push the system a little bit more and if I could optimize the system to get a higher yield of glycine production. And to do this, what we did with the help of Nitin-Bora was to add one by one the components of the pure system on top of a 5 times diluted cat cycle and measure how much glycine could our system produce. And you can see that after a few steps of optimizations, here you can see only the most important ones, we are already able to improve the performances of our system about 3 times. So in the end, we are producing about 300 micromolar glycine, as you can see in the slide. And this was way enough because we were way above the linear range I mentioned you before. And so this was the time of activating both the metabolic layer of the catch and the genetic layer of the pure system by providing propionyl-CoA for the catch and providing a DNA template encoding for GFP for the pure system. And you can see that once the GGT1 aminotransferase was present in the system, CO2 could get fixed into glyoxylate that could be turned into glycine that could be used by the pure system to produce a nice fluorescent signal for the GFP production. And I was really happy about this experiment because it meant basically make all of these enzyme components work together. But at the same time, if I look at this system a little bit from outside, what I notice immediately is that metabolism, that is the cat cycle, is producing something that is a building block, an amino acid, that is glycine, for the genetic machinery of the pure to work. But this link between these two systems only goes in one direction, between the metabolism and genetic machinery. But on the contrary, what happens in living systems is that living systems are able to self-sustain through cyclical processes in which the genetic layer is providing components to metabolism, such as it encodes, for example, DNA enzymes, and the metabolism provides back to the genetic layer nucleotides, amino acid building blocks that are necessary to process the genetic informations. And so I really wanted to see if I could make use and mimic this feature that is really typical of living systems in vitro. And then I wanted to see if I could basically build a cell-free system that could self-sustain using CO2 and taking advantage of this feature. That is, I try to make these two layers interdependent with one another. And to do this, I started by assembling a special MGLN where all components of the catch are present, all components of the pure system are also present, of course, apart glycine. But there is the link enzyme, that is the GT1 enzyme aminotransferase, that is missing here connecting the two systems. And initially, I decided to switch on only the metabolic layer of the catch. That is, I provided propionyl-CoA, and I kept off the pure system. That is, I omitted any DNA sequence into a system. And so you can see that the catch cycle can produce glyoxylate, but of course, I could not detect any fluorescent signal in the plate reader for the co-TXTL of GFP and GT1 aminotransferase. I then did the opposite. That is, I switched off the metabolic layer of the catch, omitting propionyl-CoA, and switch on the pure system by providing two DNA templates for the co-TXTL of the EGFP reporter and the GGT1 aminotransferase. But you can see again that I was basically recording no fluorescence into the plate reader. And why? Well, it's quite easy because glycine was not present into the system. I then decided to supplement minimal concentration of glycine to the MGLN that is 10 micromolar. That is the lowest point into the linear range we mentioned a couple of times before. So it's a very low concentration. And this glycine was anyway enough to produce minimal concentration of the GGT1 aminotransferase enzyme that you can now see appearing here on the sketch, and also minimal concentration of GFP, and you can see it now in the plate reader. But as you can also notice, the system goes to equilibrium quite fast after about 30 minutes. And why? And the answer is because such a small concentration of glycine, 10 micromolar, has been already consumed. And this was the time I tried to push the system out of equilibrium by activating metabolism. And so now, providing propionyl-CoA, the catch cycle could start producing glyoxylate, and glyoxylate could get converted into glycine by the small amount of GGT1 aminotransferase previously produced by the pure system, and glycine used by the pure system to produce more of the GGT1 enzyme, and this in an autocatalytic fashion. And you can now see such a nice TXTL curve in the plate reader for GFP production. And I was actually really happy about this experiment because, as you can see, we were able to extend the working time of our cell-free system from about 30-40 minutes until 10-12 hours. That means more than one order of magnitude, and this through the interdependency between the metabolic layer and the genetic layer of the system. And so after this result, what I wanted to do, I wanted to go back and work on self-regeneration, as I mentioned at the beginning of this talk, and build on the work of Barbora Lavikova and Nadanai Lacunacorn. And so I wanted to regenerate a few enzymes of the metabolic layer of the catch cycle. And so I chose specifically two core enzymes of the catch cycle that are necessary for performing CO2 fixation through catch, that are the ethyl malonyl-CoA mutase and the methyl ethyl malonyl-CoA epimerase that work in this step and also in this step of the catch cycle. And I went on basically working with the same strategy that I just presented before for the self-integrating system, where initially I decided to switch on only the metabolic layer of the catch, providing propionyl-CoA that now could be converted until methyl malonyl-CoA, but methyl malonyl-CoA could not be converted to succinyl-CoA because the epimerase enzyme is missing for this step. And I kept off the catch cycle without providing any DNA template. I then did the reverse, that is, I switched off the metabolic layer of the catch by omitting propionyl-CoA and switched on the genetic layer of the pure system by providing 3 DNA sequences for the co-TXTL of GFP reporter as well as the EPI and ECM missing enzymes. And again, I could not record any signal. Why? Because definitely glycine was missing. And then as I did before, I supplemented 10 micromolar glycine that it was used immediately by the system to produce the missing enzymes in the catch cycle. You can see them now highlighted in green here on the sketch on the metabolic map of the catch cycle, and also produce minimal concentration of EGFP as you can see on the right hand side of the slide on the TXTL plot. And but again, after about half an hour, the system stopped working, went to equilibrium because glycine was consumed. And then I decided again to push the system out of equilibrium by activating metabolism through supplementing propionyl-CoA. And now methyl malonyl-CoA could be converted to succinyl-CoA because now the EPI enzyme has been previously produced by the pure system. And then again, ethyl malonyl-CoA could be converted into methyl succinyl-CoA now because of both enzymes that were produced by the pure system. And so basically CO2 could get fixed into glyoxylate, turn into glycine, and glycine used to produce more of the enzymes of the metabolic layer of the catch. And this again in a feed-forward manner, meaning that I was in the end able to build a cell-free system where the activity of its metabolic layer, that is CO2 fixation, could be improved by the activity of its genetic layer, that is the pure system. And I was really happy about this because it meant basically running a system composed of more than 50 enzymes coming from all domains of life and working simultaneously in vitro. And I was even happier when I was able to run this system inside cell-size droplets. And so up to now, what you have seen is basically metabolism producing building blocks, that is amino acids. But as mentioned at the beginning of the presentation, basically metabolism can also produce energy. And in this regard, I think I was really happy at the beginning of my postdoc to collaborate with Shanshan Luo, also from the herb department, who was able to design and engineer the AAA cycle, that means the acid aldehyde ATP cycle, that is a new to nature electrobiological module that can regenerate ATP from electricity. And this happens with a minimal set of biological components, as you can see here in the metabolic sketch. And also, it does not require the presence of any membrane. And for this specific reason, it is very ideal to be used to power in vitro systems such as transcription and translation. And for this work, I was in charge of helping Shanshan coupling the AAA cycle with the pure system a special pure system actually that misses the energy regeneration module. And so now you can see that once the AOR enzyme, that means aldehyde oxidoreductase enzyme, that is the most important enzyme in the cycle, is present into the system, then the ATP produced from current could be used as a monomer for transcription and source of energy also for translation. But instead, once the aldehyde oxidoreductase enzyme was missing, then ATP was not produced, and in fact, I was recording a flat line into the plate reader for the TXTL of GFP. So now, we mentioned before metabolism as a very complex network of chemical reactions that is also dynamic. And in fact, it is able to replenish continuously metabolic intermediates that are driven away through biosynthetic routes. And in this regard, I think that a keynote work has been performed by Christoph Diel a couple of years ago in our department, was able to engineer synthetic anaplerotic modules for replenishing some of the intermediates of the cat cycle that were drained away for the synthesis of a polyketide. And let's say, building on this, what we are trying currently to do is to develop, together with Nitin Bor, a second MGLN system where the genetic layer can they get activated upon reaching a specific concentration of a metabolite, that in our case is glycolate. And you can see that only once glycolate concentration becomes higher than a certain threshold, then transcription and translation can happen. And this can close a loop of an aplerotic cycle that should replenish intermediates of the catch, thus extending the working time of our metabolic system that mimics what happens in nature under the name of self-repairing. And so up to now, again, we thought about metabolism basically only in the sense of biosynthesis as anabolism. But in reality, you all know very well that the living systems and cells under specific conditions of stress, such as nutrient starvation, or once some proteins, for example, are misfolded and then can become toxic, cells are able to recycle some of their components, could be protein components, into their building blocks. And so this regard, I think that I did quite a nice job during my PhD at the EPFL in Lausanne, Switzerland, where I was able to prove that short peptides, you can see here, such as magainin, glucagon, somatostatin, could be, as well as mixtures of peptides without separating them, could be recycled into very different proteins, such as fluorescent proteins. You can see here M-scarlet or GFP that has nothing to do with the initial materials, and is outside living systems and using the transcription translation in the pure. And this could also happen we could make it work also in multiple rounds of recycling. And this was not limited to peptides, as we could recycle also engineer relevant proteins, such as beta-lactoglobulin. You can see here on the top left of the slide, that is a protein from the milk industry in Switzerland that can be used, for example, to build these films that are used by the Metzenga lab at the ETH Zurich to purify water. They are composed of amyloids of beta-lactoglobulin, and we're able to recycle such protein-based materials into, again, in this case, GFP, so protein that has nothing to do with the initial material. And this, again, could also work with even more complex proteins, such as silk, that has been used for a plethora of different applications, ranging from structural applications to even bioelectronics. And as you can see, we're able to recycle silk into green fluorescent protein. And so we named this approach to recycling nature-inspired circular economy recycling, that we like to shorten as NACRE. That is based basically on three different pillars. And the first one is that polymers should be sequence defined. The second is that polymers should be should have on the backbone some bonds that should be reversible cleavable, and also polymerization from unseparated mixtures of monomers, such as in nature the ribosomal synthesis. And what is the beauty of this approach is actually that if in the future man-made polymers or synthetic polymers were to be designed and built with these features, these three pillars of Necker, then basically recycling polymers, synthetic polymers such as plastic, would basically mean transform an unknown mixture of different materials into the material of need multiple times, every time a different material, and every time virgin quality material, and is material of need by the community. And so I think that this principle is actually very, very cool. And we also proved that this is not only working with the set of the 20 proteinogenic amino acids, as it can also work with non-natural amino acids. And also with the backbone bonds that are different from peptide bonds, as we could, together with a vein allele in this case, we could recycle calf DNA into nucleotides, and then nucleotides after phosphorylation were used again as monomers to run the PCR of a different target sequence. And so after this, you have seen, of course, already in this long talk, that I mentioned many cell-free systems that are quite complex, as they are composed of many different elements coming from all domains of life. And it's really dozens of elements working simultaneously together. And if in the future I want to build autonomous self-free system, that means self-free systems that are capable of make use of energy sources as well as building blocks from their surroundings in order to remain functional by replacing and synthesizing some of their components, their own components, then we would need um, to integrate together even more components. And for this, how can we do this? So one way we are currently using is machine learning, and in the so-called active learning workflows that we couple with robotic platforms in the lab. And currently, I'm trying to apply this to optimize the strategy to optimize the decision-making system that I mentioned quickly before in the presentation. And you can see that after only 3 rounds of active learning, we are already finding some conditions where the performances of the systems are actually higher. And this is one possibility. And then there could be actually a second complementary and different possibility that could be using in reality evolution. And this is why. Because as you know, in living cells, metabolic circuits and metabolic networks composed of many different enzymes are connected, are coupled with cell growth and also cell reproduction. But on the contrary, when we try to assemble synthetic cells or synthetic protocells from non-living components through the bottom-up approach, we normally decouple the function of the enzymes from the growth and reproduction of our synthetic cells. And so one possibility in reality could be using the so-called autogene networks, where the function of some gene products of interest, such as enzymes, are coupled with expression of DNA polymerases, for example, so that basically we could create a positive feedback loop that would regenerate the system. And in addition to this, actually, we could try to evolve these complex networks by using in vitro Darwinian selection because of spontaneous mutagenesis. And so I think that this could be an alternative strategy, so to try to optimize our complex systems. And well, in addition to this, every time, you have to build new metabolic pathways, new metabolic networks. You need a lot of prototyping. And prototyping actually means injecting thousands of samples in reality into the mass spectrometer. And I was very lucky during my PhD and postdoc to work at the EPFL first and then at the Max-Planck-Institute in Marburg, where we had a lot of resources in terms of instruments and also money. But still, this process takes a lot of time. And for this reason, I was very happy last year to help my colleague Sebastian Bartel that developed this nice transcription-based sensor that I tested on the metabolism of the catch cycle. And you can see again, this could be a fast and high throughput method that could complement mass spectrometry and also allows minimal sample preparation. And it is also quite not only fast, but also cheap to use. And so after now this very long talk, I actually would like now to conclude this presentation by quickly mentioning what are my current and future research interests that I will carry out as independent group leader soon. And as you can see, as you know, there is a field in material science that is called engineering living materials, where materials are built with some lifelike properties because living cells, normally engineered cells, are embedded into non-living scaffolds. And properties such as self-regenerations and also self-lubrication, as mentioned in the slide, have been achieved. And these materials have been already applied for really many applications, ranging from structural applications, like even in concrete, to bio and biological and biomedical applications. But at the same time and in parallel, using a completely different approach, synthetic biologists over the last years started to think about synthetic cells and cell mimics for medical applications, such as, for example, tissue angiogenesis or neural differentiation. And so really at the interface between these two fields, what I would like to do in my own lab is to open a research line for developing metabolically active materials. That is a new class of soft materials that are functionalized with cell mimics, equipped with cell-free metabolic reactions. And through this, they are able actually to interact with the living cells for therapeutic and medical applications. And these materials should work in a sense, compute as well as react fashion, making use of energy and matter available from their surroundings, and using these resources to build output molecules of need, for example, a drug. And with this, I really would like to conclude this presentation by thanking, first of all of you for the attention, thanking my collaborators, starting from Toby Erb, of course, and also I would like to thank Francesco Stellacci and Sebastian Merkel, my supervisor for my PhD, and also I would like to thank EMBO for funding my postdoc, and I'm here happy to take any question. Thank you very much.
Thank you so much. That was an amazing talk. I really love this work. There is a lot of questions in chat. Can you see chat?
Yes.
If you can read a question out loud before answering, that's going to help with the recording.
Sure. Thanks, Kate. But I think, okay, yes, here I can see it. Okay. So Edward is asking, when you make delta glycine pure preparation, I guess, do you not get contamination from glycine that co-purifies with ribosomes and synthetases? Do you do something to co-purify glycine? So, well, yeah, I think, I mean, really minimal concentration of glycine can be there, but this might be below it might be really at the micromolar range or well below 5 micromolar. So we have done some tests, but we measured really small amount of glycine. So I think that can happen, but probably depends how you prepare the system. But it's a very good question. Thank you very much, Edward. And then we have a question from Anna. Great talk. Which Pure did you use? Okay, I can answer immediately. So it's the Pure Flex, and then homemade. No, this also helps also maybe to clarify on the previous question that in the end the Pure we used is the Pure from the supplied from PureFrax, so it's a commercial PureFrax. So in the end, I didn't have to purify by myself all the components of the Pure. We just adjusted a few things. We buffer exchanged the PureFrax, of course, because there were components that were not really compatible with the metabolism. As I quickly mentioned before, in the optimization of the Pure, we omitted a few small components, did a few changes, but overall, it's the base is especially the enzyme solution is the commercial PureFrax solution. And do you have the full ingredient list if it's a one, or do you rely on something like NEB system? So yes, I mean, I think if you download the supplementary information of my paper, postdoc paper that was mentioned on the slide, that was published last year in 2024, I think you should find all the details because I try to be as more precise as possible. And I assume you make your own amino acid mix, right? Yes, I definitely do that. Yes. So basically, from the pure flax, I take the enzymes and the ribosomes and buffer exchange them with solutions. You can definitely find all detail in the supplementary information of the paper. In the NECA scheme, how do you control? So here, Enric. So in the NECA scheme, how do you control what exactly is recycled? For example, how do you not digest the ribosome's own protein in the mixture? Oh, that's also it's a good question. Thanks for asking. I actually did not spend many slides on the NECA, but in reality, the steps were not done simultaneously. So I was first chopping down proteins or peptides or protein-based materials or mixture of proteins and protein-based materials with proteases. And then I was using aminopeptidase, filtering the solution, then using aminopeptidases, and then again filtering the solution, the enzymes away, and then taking the pool of amino acids obtained and spiking them inside TXTL reaction in the pure system. So I analyzed the samples, and I think that the filtering with the small filters, the chance uh, to to carry over enzymes was relatively low. But anyway, it's a good question. And great presentation, thanks, by Humbert, and really inspired science. Thank you very much. What do you use as training data for your predictive decision-making AI model? Okay, this is a good question. Thank you very much. And so what happens is that basically the approach we are using is called METIS. It has been published by our lab, so Tobias Erb lab, I think either in 2022 or 23. So you can definitely find the paper online, and it was probably also mentioned, I'm sure it was mentioned also in the slide. And this approach is called active learning, is basically on the first round gives you like almost a random uh, set of experiments of conditions to run, and then you run these first conditions in a robotic platform that is called Echo, and then you run your experiment, and then it depends what you analyze, if it's something like a mass spec or if it's something like a TXTL, and or both, like what we are doing currently. And then you feed back this data into your algorithm, and then the algorithm starts suggesting, according to, of course, the some parameters you set, suggest new condition to test for the next round of experiment. And of course, the second also is going to be probably with similar results to the first one, but as you have seen already after the third round, some things start changing, and hopefully it's going to be also the case in the next rounds. And then, wow, a lot of questions. Thank you very much. Cool science. So thanks again, Dana. Sorry, if missed that. Where is your own lab going to be? Oh, also at the Max Planck. I would love to work with you. Okay, thank you so much. I'm gonna be very happy to let everyone know when everything is set. Thank you so much for the question. I will remember. We remember this. Thank you so much. Moon, great talk. Thanks again, Moon. And Janar as well. Awesome presentation and work. Thank you so much, Janar. So as you have seen from the slide, the evolution part is something we work together with Janar and also Lynn Rothschild. And I was happy to collaborate with them to write this nice idea to try to optimize our systems in the future. And then Suzanne, fascinating work and great presentation. Could this system could be to generate synthetic cells capable of division? Hopefully. In this case, we probably would need to also contact and collaborate probably with other people such as maybe Petrashvili that would probably be very happy to collaborate, and also other scientists for sure in the world. So yes, I think that's a very good point. Inspiring. Thanks for asking. And then amazing talk. I love the view of smart autonomous materials. Can we also use natural polymers from other sources? Also, yes, of course. There is an email to contact you for further connections. Thanks. Yes, thanks, Ben. You can definitely look for my name on the MPI website, or just if you type on Google, it should sooner or later come out. So my name is Simone Giaveri, and you can find me on the website of the MPI Marburg for the moment. And you can definitely contact me and write me. I'm always very happy to be in touch, and hopefully maybe collaborate in the future. Let's see. And Kate, maybe do you have any question for me?
Thank you so much. Awesome. Now, I think a lot of the questions that I had, some other people already asked. My main one was, how are you getting rid of the residual glycine? But you talked about that already, because
Yeah, so basically, to be honest, Kate, I didn't pay so much attention in getting rid of it because I tested it and the system looked relatively clean. So I mean, when I was using 10 micromolar, this is really low concentration. So
yeah,
let's say the impurities were lower, way lower than 10 micromolar. So I agree on one side. Probably completely clean is really difficult also because I think even if the purity of the other amino acids is high, dependently on the supplier you use, but it's high. But as glycine is the smallest amino acid, I'm also not very sure if some contamination can come from that. Maybe it may also be that some tRNA that we use, because tRNA I mean, you pre-deacylate
yeah,
the tRNA, but you are never sure that this happens probably at the 100% yield. And so I'm pretty sure that it's quite unavoidable to have traces of glycine in the system. But if this is way below, or at least almost one order of magnitude lower than the 10 micromolar I was using, that is basically already quite low. So if the impurities are in the micromolar range, I guess that overall the impact of this tiny amount of glycine should be low on the overall behavior of the system.
yeah,
And also to clarify this, so once we did the interdependent MGLN, we also saw that in the case the two systems were really interdependent because we are using the DNA encoding for the correct transferase. Then even if I added different, so I doubled the amount of glycine initially, then there was no change in the final output. That means that the system are interdependent and the majority of the glycine was produced from CO2. If instead I was not producing the functional GGT1, but I was producing a DNA sequence containing the same amino acids, but a protein that was actually not functional because the amino acids were scrambled. Then what happened is that the system was dependent on the initial amount of glycine. And this is clear because the two layers were not interdependent with one another. And so I think this is one more um, scientific proof, let's say experimental proof, that even if you have, I don't know, 1 or 2 micromolar glycine contamination from amino acid tRNA, purification of enzymes, or whatever it is, then the impact of such a small amount of glycine is not really big, or actually, I would say is almost negligible on the overall performances of the system.
Yeah.
I hope I was I was clear enough.
Thank you. Yes, that makes perfect sense, especially in light of your data. That's really clear.
Thanks.
All right. I think we're at time anyway, so I just want to say thank you so much, and I'm excited to talk more about this system and see how your own lab grows.
Thanks, Kate. I'll be very happy to be in touch also for the exactly like new research and hopefully link more with the bio and biomedical field. I would be very happy to, of course, be in touch and meet in the next conference or event. Thank you very much for hosting me.
Thanks, and have a great day, everyone. Bye.
Thank you. Bye bye.